Good afternoon, everyone, and welcome to the ATMP Horizon session here at the Health Data Forum Global Hybrid Summit 2025. My name is Oliver Brown, and I'm delighted to be your host for this session. It's a privilege to help guide today's conversation on how advanced therapy medicinal products, ATMPs, are reshaping the future of healthcare. So as a quick introduction, I'm a project lead at the Life Sciences Hub Wales, which is an arm's length Welsh government funded organisation aiming to promote adoption of new innovations into the NHS in Wales and boost the life sciences sector in Wales. My work is focused on advanced therapies, and as part of that work, I joined the Advanced Therapies Wales programme meetings. This is a national programme hosted by NHS Wales with a remit that covers bench to bedside and beyond. This session today is part of the Trends House series, and we turn our attention to ATMPs and the data, systems and partnerships that bring them to patients. We'll explore how real-world evidence and bioinformatics are used for development, personalisation and approval of the next generation of ATMPs. We'll also look at what it takes to scale these therapies and sustainably and equitably, especially in underserved populations. So we've got a number of contributors today. We have a rich mix of perspectives from Wales and the UK, as well as around the world. We have a packed session starting with a number of talks, and we have a panel session at the end. So thanks everyone for joining us. Let's dive in. So our first session is from Dr. Hannah Crocker, who is the ATMP Outcomes Programme Manager at NHS Wales Joint Commissioning Committee, who'll be giving a quick introduction on ATMPs and then discussing the use of real-world data, outcomes data in advanced therapies. So unfortunately, Hannah couldn't be here today, so she's recorded her session. We are then joined by Dr. Ibukun, Aini, Falagiki, and Dr. Rachel Jemin from NHS Wales Joint Commissioning Committee, who will be able to answer any questions that we may have as well. So Joanna, are you able to put up the first video?
I'm Hannah Crocker. I'm the ATMP Outcomes Programme Manager at the NHS Wales Joint Commissioning Committee. And over the next 10 minutes or so, I'll share with you how we're using real-world data to reduce uncertainty and better understand the impact of advanced therapies in Wales. But I thought I'd start with a bit of background as to what advanced therapies are. So while the term advanced therapies can sometimes have a broader remit, today I'm referring to treatments classified as advanced therapy medicinal products, or ATMPs. ATMPs are innovative treatments that use genes, cells, or tissues, and they're often one-time treatments for conditions with few alternatives. And they aim to treat the underlying cause of a medical issue and not just the symptoms. So here I've broadly grouped them into two categories: gene therapies, which involve a genetic modification, and non-gene therapies, which use cells or tissues but without altering their DNA. So gene therapies can be in vivo, where the genetic change happens inside the body, or ex vivo, where the cells are modified outside the body in a laboratory and then reintroduced to the patient. And I'll go through these two in a little more detail on the next slide. So non-gene therapies include somatic cell therapies, such as tumor-infiltrating lymphocytes, and they're currently being developed for diseases such as skin cancer. A current example is a new treatment called Liposucel, and that could become available next year. This treatment is made up of cells that already exist in our immune system, but only in really small numbers. So for this therapy, these cells are multiplied to create a therapy that contains lots and lots of these immune cells that can target and destroy the cancer cells, so you have extra immune support to fight the disease. And finally, we have tissue-engineered products, and these are made up in a similar way to somatic cell therapies. However, they're used to regenerate or repair damaged tissue rather than treat diseases. An example is a treatment called Spherox that's used to repair defects in knee cartilage. So gene therapies are the most common type of ATMP, so I thought I'd go into these in a little more detail. So in vivo gene therapies, where the genetic change happens inside the body, generally target genetic diseases, And these are diseases where a defect in a gene has been linked to the symptoms of a disease. And these therapies are often, therefore, designed to deliver a corrected copy of this faulty gene directly into the body. And this is usually done through the use of a vector or a delivery vehicle, which is often a safe version of a virus. And that virus has then been modified to target specific cells within the body. For example, if you're hoping to treat a disease that stems from the liver, you probably want the therapy to be targeted to the cells in the liver. A good example is the gene therapy etranacogene dezaparvovec, or Hemgenix, which was recently recommended for use of treating haemophilia B. Patients with haemophilia B don't have enough clotting factor called factor IX, so their blood doesn't clot properly. Hemgenix works by providing a working copy of factor IX gene, which aims to help the blood clot properly and should have a sustained effect over a long period of time. And this should reduce bleeds, it should reduce the need for regular blood products, and therefore improving the quality of life for patients with haemophilia B. Ex vivo gene therapies involve a more complicated process, as current treatments are made using the patient's own cells to make the treatment, and this is known as autologous treatments. So it starts with collecting the patient's cells through a process called apheresis, and these cells are then shipped to a laboratory for genetic modification. The genetically modified cells are then shipped back to the hospital so they can be reinfused to the patient. A good example is CAR T therapies, which are used to treat certain types of blood cancer. And here the patient's T cells are reprogrammed by genetic modification to better target and destroy the cancer cells. So advanced therapies offer transformative potential, especially for patients with few or no other treatment options. They offer hope towards a curative outcome, which could save lives, it could reduce lifelong treatment burdens, and ultimately improve the quality of life for patients and families. But they also come with significant challenges. These treatments are associated with very high upfront costs. For example, the recent gene therapy for haemophilia B that I mentioned earlier has a list price of £2.6 million per patient. And there's also considerable uncertainty around their long-term effectiveness. We're talking about new treatments that have been developed over the past 10-15 years or so. So we don't yet have decades and decades of outcomes data to show how well they work over a long period of time. And despite this, we don't yet have a robust standardized system for collecting real-world data to understand their true impact. And this is the gap that our programme is working to bridge. So funded by the Welsh Government, the ATMP Outcomes Programme aims to move from uncertainty to insight, using real-world Welsh data to evaluate the value and effectiveness of ATMPs in Wales, and not just relying on clinical trial data from controlled settings or extrapolations from other countries, but building evidence from real patients here in Wales. We're hosted by the NHS Wales Joint Commissioning Committee, who commission all NICE recommended advanced therapies for the population of Wales, and we work closely with the Advanced Therapies Wales programme supporting their delivery plan. Our programme is made up of 3 key work streams. Retrospective data, where we're analysing existing data to see what we already have and what it can tell us. Here we have an ongoing project exploring CAR T therapy in diffuse large B-cell lymphoma, and we're hoping to evaluate its impact on patient outcomes and lifetime treatment costs. This project's not only improving our understanding of the impact of CAR T therapies in Wales, but also highlighting what data is missing. That's helping us shape our desired future data landscape. Which leads us nicely onto our second work stream that's looking prospectively at designing that desired future data landscape and identifying the steps that we need to take to get there. So looking at what data we currently have, and more importantly, what data we don't have, and then working with the clinical teams to embed this data collection into their routine clinical care. And finally, we have a third work stream that is exploring sustainable ways to fund these high-cost treatments through the use of innovative payment models. But in the interest of time, I thought I'd focus the rest of this talk on our prospective work stream and discuss our plans for the future data landscape. So as part of that work stream, we're developing an advanced therapies commissioning dashboard. And the idea is to capture prospective real-world data and including both clinical and patient-reported outcomes. This will help us build a dynamic evidence base to better understand the impact of advanced therapies in Wales. And the dashboard will also help us visualize access to therapies across the country, track patient journeys, and eventually be able to link to those long-term outcomes. It'll also help us explore equity of access and consistency of outcomes across Wales. And while most of our initial setup has involved manual data collection, our plan is to automate these processes as much as possible to minimize the burden on our clinical teams. And we can also use these initial manual data sets to assess the feasibility and reliability of a more automated approach going forward. But it's all well and good having all this data collated, but there's no point in it just being sat on a database somewhere not being used. So we need to make sure that this data is used effectively to inform our decision-making processes. And to do this, we need to embed data capture into routine care, and not just using standardized clinical outcomes, but also those patient-reported measures such as quality of life. Capturing both gives us a more complete picture of the overall treatment impact and also gives us a meaningful baseline to compare new therapies to as they're made available. But for this to be a success, we really need to strengthen our data sharing mechanisms. As commissioners, we need visibility across the whole care pathway from diagnosis and including long-term outcomes to ensure equity of access at the beginning of the pathway and also to support our value assessments at the end of the pathway to ensure that we're commissioning the best available treatments for our population that provide the best outcomes. We're currently working with health boards in Wales to update our data sharing agreements to facilitate this long-term monitoring. So once we have this data, we need to make sure we use it effectively to drive equity by identifying and addressing any variation in access and outcomes across Wales. And finally, we really need to integrate this real-world evidence into our decision-making and commissioning processes and move towards outcome-based commissioning models that reflect real patient experiences and local contexts. So diolch am wrando, thank you for listening, and thank you for the organisers for the invitation to present today. Apologies for not being there with you live today, but if you have any questions about our work, please don't hesitate to get in touch. Thank you very much.
Excellent. Thank you to Dr. Crocker for that amazing opening presentation. So we have got representatives from NHS Wales Joint Commissioning Committee here. So I'd just like to open up and see if anyone has any questions on the programme or anything they'd like to ask. Perhaps if I go first with a quick question. So have there been sort of particular challenges on working with the retrospective data? Have you identified sort of gaps in the data which have been difficult to overcome?
So some gaps have been identified, and then it has to do with the ways of reporting of the notes. Some of them might not be easy as such to extract out without the help of the clinical oversight, like some details are missing, but overall, with the help of the clinical oversight, we're able to extract enough data needed, and then the level of the missing data gotten so far is low. But yes, there are gaps in the data being extracted, and it can be better.
Excellent. And you mentioned as well the sort of data moving forward and some of the barriers there. What sort of barriers have you found with collecting or setting up the systems to collect that data moving forward?
I'm not sure of what you mean by the barriers in setting up the system.
So has it been a simple process to start collecting that data up front, or have there been any sort of issues? Has there been good clinical buy-in for collecting this data moving forward?
I think going forward, I think it's going to be easier collecting the data prospectively going forward, instead of having to go back into getting the data and extracting them. Some missing spaces might be in between, but if it's prospectively, the data will be available, and then you can get the information and all needed from the patient at once.
Rachel?
Yeah, just to come in and add. So I think that the work that has been done as part of the retrospective piece has really helped inform sort of what are the areas we need to concentrate. And we've had really great clinical engagement and buy-in. So actually working together with the clinical team to sort of plan for this prospective work. So I think that has really helped. So until we start, I think it's going to be really hard to know where those gaps are. But obviously, you know, we face the same challenges that people will see around the governance, around the systems, around how do we reduce duplication, because what we want is to be able to offer an efficient system where we're not duplicating and we're not asking for people to put in extra time. So we've been navigating all of those to try and make it as streamlined as possible. And I think having that clinical engagement and buy-in, and obviously the fantastic support that Ibukun and Hannah have provided, I think that has really helped to move things forward. So hopefully we'll be back here telling you about the results from that piece of work next time.
Excellent. Any further questions from the floor? No. Just wondering if you're able to expand on what sort of data. Oh, sorry, Tiago. I'm sorry, we're having difficulty with your sound. It was cutting in and out.
Sorry, it's not here.
I changed my microphone now. You cannot hear me now.
Yes, we can hear you.
You can? Okay. So I'm curious about the numbers here, right? Because for this type of treatments, it's so expensive that the numbers end up being small just by definition, right? And so to follow it longitudinally, I'm curious if you have any idea of the numbers, or you just follow the ones that you have and then you make the best out of it, which is what we do very often as well, right?
Yeah, so because of the criteria to make patients eligible for these treatments, and obviously the numbers are quite low, so we will be looking to include all of the data that is available. But yeah, we have low estimates. It depends on the treatment that they're receiving.
And also not talking about automation capture, right? I mean, the LLMs and audio immediately the notes or that type, is that, I guess, what you're exploring when you're talking about automation of this data capture?
It's around how we can collect the data once and use it many times. I think it's one of the things that we're really passionate about doing, and trying to reduce that sort of admin burden on the team so that we can utilize, and also that we can feed in sort of the information that we can extract from that data back to the teams to complete that loop.
Excellent. I think in the interest of time, we will move on to our next session. So next we have a couple of spotlight talks, and first up we have Dr. Andrew Roddam. So Dr. Roddam is Chief Strategy Officer at Everyone Medicines and the Chair of SNOMED International's Management Board. He's a renowned epidemiologist with over 25 years experience. Joanna, if we could load up the slides, please.
I'll probably share from here.
You've got it. Okay.
Perfect. So thank you, and thanks for the opportunity to share a little bit about what Everyone Medicines is doing in this sort of framework of advanced therapies. And really what we're trying to do is think about what does it look like to create these scalable solutions when you're developing therapies which ultimately will only treat one or a very small number of individuals. So very, very different from the traditional paradigm of drug development. Let's just go forward. So what are we working in? So Everyone Medicines is really focused on these very, very ultra-rare genetic diseases, or diseases that have a genetic cause. Most of them are presenting in childhood. Almost none have any sort of approved treatments that are really changing disease course. Most of the treatments for these rare conditions are to reduce symptomatology. Broadly, it's about 50 million the age 5, and much, much more die before they reach adulthood. So this is a really difficult situation and a difficult group of children to potentially treat, but with a huge opportunity if we're able to think about how we develop medicines which will solve some of these challenges. And really where we are is that we've got probably the scientific technology to think about how we treat these patients. And I think that sits in the realms of gene therapies, ATMPs, cell therapies. There's a whole raft of treatments. We're really focused on antisense oligonucleotides, which are in the RNA category. So they sit somewhere between cell, gene, and small molecules, but they're targeting the RNA transcription so that they're blocking the abhorrent production or reduction of proteins. And the challenge that we have is not so much the scientific one as you often face when you're developing drugs. It's very much the population that we're trying to treat is far too small to go down the traditional sort of regulatory approval pathway. If we're treating 1, 2, or 3 people, it's not clear what that regulatory pathway looks like. And the work of Everyone Medicines was really started on the basis of our one of our founders, Julia Vittorello, whose daughter Mila at age 6 was diagnosed with an incredibly rare form of CLN7 Batten's disease, which presents with seizures, loss of mobility, and general rapid deterioration. Julia got connected to Tim Yu at Boston Children's, and within the space of 12 months, Tim had designed and got approved as for compassionate use by the FDA, an individualized drug, which he named Milacin after Mila, which was given to Mila just after 12 months of him first seeing her and being able to work on the sequence. The drug itself created a significant improvement clinically for her. Seizures reduced, she gained back mobility, she was able to smile again and interact with her mum, and actually stayed on drug for about 2 years, by which time she continued then to start to decline again. The damage that she'd already suffered neuronally was too significant for the drug to reverse any of that. And sadly passed away at age 10. But Julia, who is a true inspiration if you get to meet her, sort of made it her life's course to think about how do you bring children these drugs in the future so that ultimately children and parents don't have to suffer knowing that there's a potential treatment but not being able to access it through the classical and standard channels of medicines as we know them today. So our vision is to think about, when do symptoms start? How do we use genetic sequencing to identify these variants, which are the causative variant, and then develop and deliver a treatment, hopefully within 6 to 12 months, to be able to, of discovering this new variant, to match the patient, directly match to the patient's cells that are causing those diseases for that child. And we're really focused on a couple of things. We know that there are 2 sort of main areas where ASOs have a potential mechanism of action. One is we get abhorrent exons, which cause sort of enhancing our protein. We need to enhance the protein expression. So we need to end up doing sort of what's called splice switching. So we skip a particular exon, which is the poison exon, and then recreate the normal levels of protein. There's also RNase-ish dependency, which are sort of allele specific, where the child only carries one of the 2 alleles, carries the variant mutation, and that uses a different mechanism to cleave where the target RNA is there to remove the amount of expression of protein, which could be toxic. They're the 2 things that we're really going after, and we're trying to pick the easy things to work on initially so that we can work with the regulators on what does this look like as it starts to come through in a regulatory pathway in the future. So if you think about traditional drug development, and I've worked in big pharma companies where 5, 10, 15 years is a normal path to go from initial concept to actual licensed molecule, ASOs, and particularly in the individualized setting, are somewhere around 2 years at the moment. So roughly from when you first meet the child or first get the cells from the child or the sequence from the child to when you're able to get them recruited into a clinical trial is a total period of about 2 years from suddenly our first experiences. And we believe that over time that could reduce even further by showing some redundancies that we currently have in the process that could potentially reduce that time frame in the future. We know that the landscape, thinking about regulation and the regulatory path, and it's nice to have Michelle here because she can add some colour to some of this, is, you know, actively thinking about how does it adapt to this sort of novel paradigm. So, you know, the MHRA has to be complemented on the work they've done on highly personalized medicines now, with the particularly starting in cancer immunotherapeutics, expanding that group to include rare disease experts, and really trying to tackle some of the challenges in rare diseases, not just for individualized, but much more broadly about how it is such a challenging area to work in a pharmaceutical setting. And it's a real sort of positive step. And we know in Europe, with the general pharmaceutical legislation, which is in tripartite at the moment, there is paragraphs included in that legislation which talk about platform marketing authorizations, where you keep one part of your medicine fixed and you change it a little bit time on time to direct it towards a new individual. And that will be equally really positive as time goes on. And the FDA, given all of the administrative changes in the US, is making positive noises in a similar direction. So I think we're quite hopeful for that. And what Everyone Medicines has been doing has been looking for patients that might benefit from this treatment. And we've agreed with the regulators in the UK and in principle in Europe that carrying out a multinational clinical trial where we select multiple patients with different variants and design different ASOs is going to be the best way to demonstrate the evidence of how this comes together. Initially, our focus is in the UK. It's at Great Ormond Street in London, and that's where our first patients are coming from. But we're also trying to establish a collaboration with the Telethon Institute of Genetics and Medicine in Naples, who are equally have a number of patients who could be eligible here. So we're looking at how do we build this evidence package of the process that we're trying to do being safe and hopefully effective for treating these children in the future. So we have this blanket master basket, and no one quite knows how exactly to describe this as a clinical trial protocol, where essentially we can recruit multiple patients with different ASOs, with different symptoms and different variants and different diseases, ultimate diseases. Hopefully in the next few days, we'll hear about the first patient that we've designed the ASO for Great Ormond Street. They're a patient in lysosomal storage disorders, and we're very hopeful that that will lead to a clinical treatment either late this year, but more likely early part of next year. And we're now working up some of the preclinical science on the second, third, fourth patient that hopefully will be able to move forward underneath this sort of umbrella protocol that we filed. So just to summarize, I think individual medicines now are a reality. They're a reality in the non-commercial world, where individualized in the US with multiple organizations and academics thinking about how you do this under compassionate use. We're really pushing the frontiers and boundaries around thinking about a business model that makes this scalable and sustainable. We know, actually, oddly, for having worked in drugs for a large number of years myself, the technology is actually the easy part here. The harder part is kind of getting the regulatory, the patient finding, the reimbursement, all of the supporting packages that we need to be available. And it's really great, the sort of work that the Wales and the Life Sciences Hub are doing to kind of try and advance these ideas in ATMPs. And we know that this is going to be inevitable, and we hope that ultimately by working in partnership and working with regulators, with governments, with health authorities, we'll be able to develop and catalyze the scalable and sustainable solution for everyone in the future. And with that, thank you very much. You're on mute, Oliver.
Thank you. That was a very interesting talk. There's some unique challenges there for these ultra rare conditions, and I think it'd be really interesting to perhaps unpack some of that in the panel session a little bit later. Thank you. Our next session is being presented by Dr. Tiago Magalhães. He is Director of Bioinformatics at M42 Abu Dhabi. He oversees a bioinformatics team who produced and analysed the Emirati Genome Project. Tiago has an extensive background in bioinformatics and genomics, and very excited to hear this talk as well.
Okay, so I'm now trying to this unfortunate event. I used to use Zoom and I could not work with anything else. And now that I'm using Teams, I cannot work from, okay, here now. Can you see me now?
Yes, we can.
Great. That was not too many seconds of embarrassment on this very stressful moment of starting a presentation on this type of Teams. Anyway, so I'm going to talk about work that we are doing at M42 in Abu Dhabi. and we have interacted with everyone medicine a little bit, trying to identify those patients, which there was none that was immediately available, but we will definitely go back. I mean, I think the work that they are doing is amazing, and it's really. I remember being in the ASHG first time that this was presented some years ago, and the whole audience was kind of absolutely extraordinary how we could move forward on this direction. Anyway, so this is a bit of M42. It's a very large organization. I'm not going to go into detail, but it has a lot of hospitals. It has a very large genomics capabilities. It has a CRO, and it has also EVS for environmental services. It's based in Abu Dhabi and interacts a lot with the ecosystem here. And of course, we partner and try to be a partner around the world for everything that relates to healthcare. So this is a very busy slide. I'm not going to go through all of it, but it's about the Emirati Genome Program, a bit of an explanation. So the idea here is to sequence all the 1 million Emiratis and for better healthcare. So that's the main aspect of this program, is that it is, of course, directed at impact in healthcare. It is very unique in the sense that it sequenced the whole population of a country using several technologies. It has a strong component of long reads that for people around the more the bioinformatics aspects is a different technology that gives you more structural variants and more aspects of what could be areas of the genome that are not well covered in the current more common short reads. And then it is an interesting population because it has gone through a rapid expansion, which means there is quite a few founder effects. And also because it's tribal and because there is very large families, there is a certain aspect related to consanguinity, which makes it more powerful in terms of discovery for genetic variants, because you just have more of specific rare variants, which makes it just easier to identify in a population. So currently we have 750,000 genomes sequenced, which makes it, I think, the largest PopGen in the world, and very large long reads and very large methylation as well around long reads. So M42 is the operational EGP entity. The data belongs to the Department of Health, which is the government of the UAE. And this is just a picture of the Illumina and ONT sequencers large that we have here in the lab. I'm not going to go into details of this, but the idea is that there is a sequence once, use all your life strategy that already was mentioned here, the idea that you collect once and then you use several times. That's, of course, the idea. Simply put, of course, you want to have the whole genome of the population in a database that can then be used in many programs. One of them, premarital screening, pharmacogenomics, HLA registry, and rare disease testing, of course, and genomic screening around the population. I'm going to focus maybe in one particular aspect where some details of how we can go and discover new variants on a disease which is blindness, which Wexterna being the first case, I think, that was regulatory and legally approved as an ATMP, and so we proceeded to do, and this is in the last stages of reviewing to send for publication, pursue on identifying all the variants related to IRDs, to inherited retinal disease, in the whole population. So because we already have covered 75% of the population, we are looking into all the known variants in all the known genes, and then we identify everyone from those 750,000, and because then we have the phenotype for these individuals in the form of electronic records that have been collected for 10 years, so we have a sort of a longitudinal aspect of this electronic health records, then we can go and we can identify whether indeed those variants and those mutations are causing blindness. As we all know, many of these studies start on the sick people, in the sense that this is a cohort of sick individuals that is being identified, which means that there is a strong ascertainment bias, because those variants are the ones that people are seeing. They are not exactly what is covered in the whole population, and this was what we believe this study is telling us. So the design is then simple. You have the 500,000 genomes. We did this on a smallish at the time data freeze that we had. For them, we have 450,000 electronic health records. And then, of course, we have families, because from the fact that we have, in this case, would be 50% of the population, we can, just by the fact that we are sampling so much, we start to be able to get families from the genetics, right? And so we map this. We have 51,000 families within our our data set. And so then you can look not only on the population level, but also on the family level. And we call this FemPop, not only PopGen, but also FemGen, FemGen, because it's family, not only population data set. Again, a bit of the experimental design. So we go into the electronic records, you go into the ICD-10 codes. We are using also LLM to understand whether not only the codes, but also from the whole corpus of each electronic record that has many visits, whether you can get a label for ERDs or blindness. And then you go to the population, and then you go for different panels that we have looked at. ClinVar, of course, being one of them, and we used one panel that was curated by a clinician that we work with, and he has curated a population-specific data set, so we look into those, and then we find these variants. And this is all the variants that we are looking, and we find some of them that are, let's say, about 0.5, which is penetrance that we believe indicate a very strong penetrance of these variants. And surprisingly, at least to me, some other people were not surprised, and I comment for my team, I mean, and I was like, okay, you are much smarter than I looking into this, because there is quite a few variants that are zero penetrance, meaning that these data sets, even at the higher level of ClinVar, 2 stars, etc., are indeed not showing penetrance in this population. Maybe is that these variants are not causal, so we were actually looking into text SNPs and not actually causal variants, but there is clearly quite a big chunk of variants that are at the zero penetrance level, which of course should not be used in any genetic screening. So looking into the family structures, this enabled clinical stratification, which then allowed us to look for novel variants, but by picking families or more than one person had a particular penetrant variant, that allows us to just believe that there is a stronger, richer signal there. And so we focused on this 76 families where they had more than one individual with this specific phenotype. And then we indeed identify quite a few variants. This is one of them in the ABCA4 variant, and this is in this PBS 12, where 4 individuals in 2 different families we could identify, and we believe that we have identified a new novel variant that is highly penetrant, and that explains this phenotype for these 2 families. So just very quickly, putting it together, how can this ATMPs be linked to EGP or to very large PopGen? I think precision clinical trials would be very obvious. Identifying rare disease and families impact action, which is what we try to do with Andrew and Everyone Medicines, is identifying individuals that could immediately be fast-tracked into any kind of these advanced therapies and very precise therapies. On the other side, which is in the beginning, right? New variant discovery, new gene discovery, and GWAS-FIV was on the pre-discovery side. In a way, these programs, funny enough, gives you the two extremes, in the sense that gives you the first extreme of the new discovery, the pre-discovery, and gets you the last extreme, which is finding patients for clinical trials. And on those extremes, so to speak, of this range of drug discovery, I think these programs are bringing a lot of value into this. I think I might have expanded my 8 minutes for a couple of minutes. Apologies, but I'm finished. Yeah, thank you.
Excellent. Thank you very much. That's, again, very insightful, interesting talk there. We're closing with a panel session. So as well as Tiago and Andrew, we are joined by 2 additional panelists. So we have Dr. Michelle Lamb and Linda Galbraith. So Linda is a passionate patient advocate with lived experience and a strong leadership role in health research and innovation. Currently serves as the PPI chair for the International Alliance for Cancer Early Detection, PPI lead for RadNet Scotland, co-chair of the Scotland Cancer Centres, and member of the Flatland Patient Voice Panel. Michelle is a medical assessor for the MHRA in healthcare quality and access, and a neuroanaesthetist by background, and has also worked in the Innovation Accelerator team at the MHRA. So thank you everyone for joining us. I think maybe if we start by discussing perhaps Andrew's point, which he brought up on regulatory path in the sort of ultra rare disease space. And I was wondering, Michelle, if you were able to sort of expand and give your perspective on that.
Yeah, sure. I mean, I think there are a few things that we definitely acknowledge that there are challenges and there are gaps in the regulatory system to facilitate this access of safe and effective treatment to our patients. So the first thing I think what I wanted to say is the classification of ATMP itself. For example, everyone medicine is developing antisense oligonucleotide. And if you look at the current regulations, that actually doesn't fit into ATMP. And it's the same for the mRNA immunotherapy for cancer. If you've heard of it, there is a recent draft guidance for these therapies. And looking at the classical regulations, it doesn't fit in. But we know that the aim of these regulations really is to provide that risk proportionate approach. So that's one thing that as the regulator, we are actively looking into. We are reviewing the regulations, and we want to make sure that the regulations follows the science and it is fit for purpose. So their ongoing work with a Center of Excellence called CERSI Advanced Therapy. So I think if you watch this space, there will be some publications soon on how we are reviewing current classification. The other challenge, I guess, is more particularly, like you said, for ultra-rare disease, even N of 1. And I guess it is all about the difficulty in generating that evidence. So like Andrew said, there are trial designs which allow starting with very small patient number, grouping them together in a master protocol. But ultimately, when it comes to applying for a license, how do you generate that evidence on safety and efficacy? It's very difficult when you only have 1 or 2 patients. So we have actually just set up a initiative called the Rare Disease Consortium by the MHRA, and we're actively working with a lot of stakeholders, including patients, industries, academics. We're closely working with our partners, so NHS England, NICE, to look into how the regulatory framework can be shaped in the future for these kind of very rare diseases. Again, I can't say too much, but in the next few weeks, we're going to have a publication. So it's the right time to be doing this panel. We will have a lot of news on this.
Excellent, thank you. And Linda, I was wondering if we could come to you and sort of whether you can provide a patient perspective on some of these things and how patient groups can work with regulators and companies.
Yeah, I am a patient with an ultra rare disease, so it's probably quite fortuitous that I'm here. I've got an ultra rare cancer. I think I'm the only one in the UK with this particular cancer, and I'm also a member of the consortium that Michelle was talking about. I've seen ATMPs increase over the last few years. I've been in an ethics committee for quite a long time, and I have seen interest in this, and I was also on ILAP on the pilot stage for the fast tracking, getting things through. The other thing that I found I decided to, because I couldn't find anyone else with what I had, I actually joined Flatiron, which is data analysis of cancer. And I think it was partly as well because on an ethics committee, I continually saw drug trials that didn't reflect the population I lived in. And I felt it was much more helpful to have real world data, but also if they were to ever find another one, it would be in a multinational project that had lots of patients. So I suppose it's influenced my choice of the kind of things I do. And I am very supportive of these developments, particularly since so many ultra rare diseases affect young and sometimes very young people.
Yeah, absolutely. Thank you. And I think you raise an important point about different populations. Sort of open question to the panel. As we're talking about sort of personalized medicine and genetic data at a population level, there are obvious risks in sort of global inequalities for this. I'm just wondering how we can sort of help support underserved populations in sort of ensuring that this data is used across the world, not just in a few select countries.
Maybe I'll start with a couple of thoughts from partly my current day job, but also my last job. I was chief exec of Our Future Health, which is trying to create a 5 million person data resource with genomic data in the UK. And one of the challenges is very much, as you said, there's, if you look at whole genome sequencing in particular, the vast majority of whole genome sequences available in the world today are in white populations. The Middle East is an exceptional point of diversity by sequencing the Emirati populations, and similarly in the Qatari and Saudi. But we've got to get to a place where it becomes routine that as part of clinical practice, particularly for rare disease patients, that they get access to the sequence and the testing necessary to be able to diagnose them. And I think particularly if you look at rare diseases as a whole, the statistics of 5 to 7 years of an odyssey journey as you bounce between services in the healthcare system, no matter really which country in the world you're in. While they try to figure out a diagnosis, often multiple rounds of genetic testing to get you there. So initially panels, and then exomes, and then genomes, and then the challenge of actually not having active treatments at the end of that the genome is driven towards. So I think there's a sort of self-fulfilling prophecy. Hopefully, as we start to get more genetic-based therapies available, genetic testing will become more prevalent because it'll be a sort of companion-like thing that has to happen prior to the therapy being prescribed. So the hope is that that's there, but I think it will take a lot of, lobbying, advocacy, the voices of the patients themselves, sort of campaigning to make sure that they're not forgotten and that they actually do get the diagnosis they deserve in a timely way, so that the treatments that are being developed are available to them in the future.
Excellent, thank you. Linda, did you want to come in?
Yes, just to talk about one of the projects I'm involved with, which is the early diagnosis, and is a global project. We, at the moment, are very much Western world expensive first-class universities, but we're moving away, hopefully in the next few years, to look at middle and lower income economies and what can be done there as well. And one of our themes is inequalities in healthcare, and that has been challenging because a lot of our universities are in the US, and wording is everything these days, and some particular universities we work with have had a pretty hard time over it. So it's seeing how we can go ahead and do what is decent research and the kind of research that we need with the environment that at the moment doesn't support it.
Yeah, thank you. And another thing which was mentioned earlier is the differences in regulation between, say, the UK, Europe, and the US. How much sort of discussion between those bodies is happening, and what can be done to sort of try and streamline that process?
Yeah, I can answer that. I think you're absolutely right. In the question about inequality, that's also something to be considered, is that harmonization with other regulators. So the MHRA has been in we have bilateral or just conversations with the EMA and the FDA, but we're also in bigger groups such as the ICH, which is the International Council for Harmonization, and there's a group called ICMRA, which is the International Coalition of Medicines Regulatory Authorities. So these are groups where we have FDA, MHRA, EMA, regulators from other countries, and we really sit together with the hope that we can set some guidances which will be applied in principle for more than one country. And that really is important for developers, innovators to develop their product. And in terms of ATMP as well, there are special working groups. So we discuss on topics, and we hope to generate more guidelines in the future to give more clarity, I guess, and also help. For example, if we talk about rare diseases, really when you look at the patient number, it's very small in one country. But when we look at the global picture, there are more patients for us to generate evidence. So definitely that having that harmonization is very important.
Excellent, thank you. Tiago, did you want to come in?
Yeah, I mean, this is of course not necessarily on my own technical skills, but on my, observation of this field and the world. I mean, I think it's hard, to be honest. I mean, on one hand, we want regulation, of course, because that saves us. On the other hand, regulation to this level makes it very hard to get patients, right? On the one hand, we want, every time you're thinking about genotyping people or doing whole genome sequencing in Western populations, it's really hard, and the people really do not want in many ways and are very against being sequenced, right, in many populations in the world. We want equity, but you look into CAR-T, it can't be cheap, right? You need to take those cells, generate, put in them the layers, inject. I mean, yesterday there is this Huntington startup I saw somewhere in the news. You need to do brain surgery, inject the cells, because it's just very, I don't see, to be honest, it's many things at the same time and going to many directions, right? If Andrew needs to fight like this incredible fight and doing one, and the regulation is still, like Michelle saying, it doesn't even fit. I don't know. I think we are really as a field and as a society very far away from dealing with all this. I brought no clarity whatsoever to this conversation, but since we are not so many, I just felt that maybe we could, I don't know, brainwave here a little bit. But I can see it coalesce, to be honest, how this is gonna go right now.
Absolutely, certainly a lot of uncertainty. Michelle, did you want to come back in?
Yeah, I think like when Hannah started, a lot of ATMP therapies, one of the biggest challenges is that cost. And as the regulator, we recognize that it's very important to make this regulatory framework a financially sustainable pathway for industries, and that's why we're constantly engaging with them. And I think with our consortium work going on, we listen to industry's perspectives, and I think opportunities like how you can leverage prior knowledge, for example, what people call a platform technology. If you can produce the first one, but the second one or the third one, it will allow you to use that knowledge and hopefully streamline some regulatory process. I think that kind of model may help with reducing the cost, but of course, we always welcome industries to talk to us, engage with us, and I think we really want to work together, because there's no point developing a framework or a license that actually isn't going to be useful or helpful for industries.
And I think as well, as the sort of population genetic data becomes more sort of available, the sort of forecasting of the expansion of treatments and the sort of cost will become more and more important. I'm afraid we have run out of time now, but thank you everyone for your contributions today. We've had some really excellent talks and some great input from everyone. So thank you very much for joining, and I'm sure these conversations will continue. Thank you very much.
Thank you.
Everybody's looking for a healthcare system they can trust. In the age of AI, transparency and ethical data practices are revolutionizing healthcare. By ensuring that data is handled with care, we build trust among patients, healthcare professionals, and stakeholders. Why does this matter? Ethical AI ensures that every decision made is aligned with human values, providing accountability and eliminating bias. This means better outcomes for everyone involved. But it's not just about technology, it's about trust. When patients know their data is safe and used ethically, they feel more confident and engaged in their healthcare journey. Let's embrace transparency and ethical data practices, because a trusted healthcare system is a better healthcare system. Transforming healthcare, one ethical step at a time.
Welcome to the future of health innovation. Are you an innovator at the cutting edge of social impact and health technology? Then get ready to discover a unique opportunity. We are thrilled to introduce the Social Tech and Health Innovation Village, a dedicated space designed for visionaries like you. This exclusive showcase is a key highlight of the Health Data Forum Global Summit 2025. So what exactly is the Social Tech and Health Innovation Village? It's a unique showcase area specifically curated for innovative companies, dynamic startups, and groundbreaking projects. This village is where social impact seamlessly intersects with health technology, fostering collaboration and progress. Curious if the Innovation Village is for you? We're calling all social tech startups, health AI, and data innovators, and real-world evidence enablers. Also, patient empowerment platforms and care pathway disruptors are highly encouraged to participate. And of course, digital inclusion pioneers who are bridging the tech gap in healthcare. Why should your innovative project be part of this village? Gain unparalleled visibility among influential healthcare leaders, policymakers, and fellow innovators. Enjoy direct networking opportunities with summit speakers and key sponsors, fostering invaluable connections. Your company will be prominently featured on the summit website with your logo and project bio, boosting your online presence. You might be wondering, where exactly can you find this exciting hub of innovation? The Social Tech and Health Innovation Village is strategically located in the President's Room, right in the coffee break corridor. You'll also find a presence in the main lunchroom, ensuring maximum foot traffic and engagement. Plus, your presence will be highlighted on both the official event website and the printed agenda. Don't miss this incredible opportunity to amplify your impact. Join the Social Tech and Health Innovation Village today.